pleased to introduce Kiho Cho, who has been working in the Department of Surgery and with me especially for 22 years. Incredible scientist, but also a veterinarian from the start and a virologist. Been a great partner for me and for the department for doing real molecular biology, and he's also got he goes into new concepts and makes you think about new concepts. So today he's going to continue with that process with his talk on the Assumptions Project. So Dr. Cho.
Thank you, Dave. So I've been studying why different people respond differently to same type of stress. In this case, burn injury, infection, maybe the mental stress. So during the course of this study, I realized that the nature is a lot more complex than I thought. From the study of finding the reason why we respond differently to same type of stress, I realized that nature is a lot more complex. This is one example. Assuming that, I'm using the word assuming that there are 3 billion nucleotides in human genome. We are looking at 4 to the 3 billionth level of complexity here. And at the same time, there are about 7 to 8 billion people on Earth. That means there are about 7 to 8 billion frames of 4 to the n system. And among that population of humans, we share the frame. That means certain nucleotide and positions are selected to make human species. At the same time, our DNA or genome information system is dynamically changing based on time and based on space. And also we see the evidence that this DNA or genome information system can form secondary and tertiary structures. So therefore, every nucleotide and their positions are important. So when we are looking at 4 to the 3 billionth level of complexity, that complexity is high. So during the course of the study, I was able to collect some evidence showing that some popular assumptions in biological science, including biomedical science, are too simplistic to decode that level of complexity. I'm not talking about just the complexity of the genome, but all the phenotypes that we see in nature. Some assumptions might be relatively minor. The other assumptions are critical because they are the foundational assumptions in biological sciences. And we rely on those assumptions to advance our knowledge in biology as well as medicine. So the question is, are those assumptions sound enough or not? If not, we might be in big trouble, like the Millennium Tower in San Francisco. So a couple of years ago, I conceived the idea of developing a project. I named it the Assumptions Project. The key goal of this project is that I like to evaluate sustainability of scientific assumptions as a group, instead of just me evaluating those assumptions, but I like to form a group of scientists and the public so that we can evaluate whether those scientific assumptions are sustainable or not. At the same time, I like to advocate for accuracy and open discussion about assumptions and related science. NIH keeps talking about reproducibility of science, but they don't talk much about accuracy. But with this Assumptions Project, I like to focus on accuracy and open discussion with assumptions, which are the foundations of key scientific studies. One of the key components of this Assumptions Project is that assumptions assay. So recently, I was able to finish the first draft of this assumptions essay. So the title of this essay is Foundational Assumptions in Biological Studies, Applications and Knowledge. How sustainable are they? So there are two chapters in this assumptions essay. The first chapter talks about assumptions of system. And there are 10 subsections. So today I'll just introduce up to assumption section A9. And I'll skip A10 because I have limited time. But with the second chapter, I'm talking about assumptions of tool. For instance, the first section, B1, talks about the expression analysis by measuring messenger RNA. All the tools that we use, are they really sustainable? And the second section will talk about the expression analysis of proteins. Are they really sustainable to produce useful data set? But since, because of a limited time, I'll skip the majority of these assumptions tool section. However, if you look at the B45 highlights, since the coronavirus is kind of very popular topic in news media nowadays, so I'll be using coronavirus infection as an example of whether the nucleic acid-based diagnostic system is useful or not. So I'll talk about 9 sections from chapter A and also 1 section from chapter B today. So first 2 sections, I presented this about 2 years ago in this forum. But I'll give you kind of the summary of those findings. There is no data, but I'll give you a summary of these findings. So the knockout mice and mutant mice, I'm talking about natural mutant mice. They are very popular for biological studies. But the genetics of those mutant and knockout or engineered mice are quite uncertain. First of all, there is a strain identification system for different mouse strains. But this current system can only work when all mouse strains share 99.9% of genome sequences. And at the same time, we have a completely sequenced reference genome. However, as of today, there is no single strain of single mouse strain completely sequenced yet. That means we don't know whether different mouse strains will share 99.9% of the genome sequences. So current strain identification system cannot work because that system does not have those 2 assumptions intact. Second, in case when you do knockout, typically knockout can target 1 exon of a gene. Let's say you target exon number 2 or exon number 4. If you assume that that specific gene produces 1 or 2 messenger RNAs, that might be okay just to knock it out. However, a single gene can produce unknown number of transcript or messenger RNAs. So if you knock out exon number 2, you may knock out some of those transcripts. However, there might be some intact transcripts which do not have exon number 2 or exon number 4. So that means if you are knocking out a exon number 2 for a gene A, you are knocking out only a subset of messenger RNA population, not the entire gene. So instead of complete knockout, it will be a partial knockout. And at this point, we really don't know how many messenger RNAs are produced from a single gene at this moment. And also, the genetic landscape of the control strain and knockout, they are different. So obviously, they are different at the knockout locus because they knocked out. But at the same time, when we are looking at the genomic landscape, they are different. That means these controls cannot really serve as controls. what's really interesting is that there is no completely sequenced mouse strain, but yet they claim that they are supposed to be identical without checking. So this is one example. Actually, we used CD14 knockout mouse from Jackson Lab. So it has two genetic background, 129 strain embryonic stem cell, and then they use black 6 as a back-crossing strain. This knockout should be CD14 co-isogenic. Co-isogenic meaning that two strains are only different at that one genomic locus. However, based on our study of these genetics of the CD14 and black 6J, we realized that they are completely different in terms of their genetic profiles. So therefore, instead of being a co-isogenic, I like to say that they are a hybrid of 129 and black 6J. If they are hybrid of 129 and black 6J, instead of looking at the phenotype of that specific gene, now we are looking at the phenotype of hybrid. Then that data cannot be interpreted in any way in terms of studying the function of the gene. So natural mutant meaning that these are not genetically engineered. However, we did survey for the genomic landscape of one mutant and its control. In addition to that mutation that was described, we saw that those two strains, one is mutant, the other one control, they have completely different genetic background. Therefore, these studies, which use natural mutant, we need to reevaluate to see whether all the studies focusing on gene function using these mutant can be sustainable or not. So when we have knockout one natural mutant, we see the phenotype. So now we are not so sure whether it is coming from that specific gene that we knock out or hybrid of two strains during the course of making these mutant knockout strain, or the hybrid of different genetic profiles of control and natural mutant. We all learned that inbred mice are almost identical in terms of their genetics and phenotypes. What is interesting is that nobody confirmed that they are identical. They just assumed that. They assumed that they are identical, 99.9%. However, on the left, you will see that that is the current assumption. The inbred mice, they are almost identical. They are different here and there, very minor. However, based on our study of inbred strains, now we realize that inbred mice are not really identical. And also their DNA is changing over time and space. Even a single black 6J inbred mouse strain, if we take DNA from the heart, that DNA is different from DNA from skin. So we need to reevaluate whether our assumption of the inbred mice, for instance, 99.9% identical, we need to reevaluate that. In criminal justice system, they are using DNA evidence. And also last year, there was a very famous case. I guess it started with Golden Gate. Anyway, so using the database from the ancestry tracking system, they said they identified the person. So I'm wondering whether the current system, DNA or genomic profiling system based on short tandem repeat or single nucleotide polymorphisms are useful or not. I show this slide at least a couple of times in this format here. In 2013, NIH has a 10th anniversary of completing human genome sequence. 2013, that means they finished 2003. As of today, so this data is as of June of 2019, but as of today, NIH couldn't finish sequencing of a single chromosome yet. There is no completely sequenced chromosome in NIH's database. I'm talking about single reference genome. However, similar to the mouse strain identification, to identify human DNA, there should be two major assumptions. One is humans should share 99.9% of genome sequences, and there should be a completely sequenced reference genome for that. We don't have either of them. Without those two assumptions intact, current DNA identification system cannot be used for identifying a person. If we are looking at the conventional genes, those genes that we learn in college and medical school and so on, we are highly homologous at 99.9%. However, if we are looking at our genome is really diverse or heterogeneous. At the same time, within an individual, our DNA is changing over time and space. Currently, the vast majority of biological science, including biomedical science, focuses on the function of conventional genes in understanding normal and disease biology. Is it adequate to collect useful and accurate data for advancing our knowledge in science and medicine? On your left, if you are focusing on conventional genes only for your study, your system is rather simple. But if you focus on all genetic elements in our genome information system, including the dynamic nature of our genome, time and space-dependent changes, it is rather complex. So at conventional gene level, individual A and B, they are very similar. However, when we are looking at all genetic elements together, they are so different. That makes sense because there are about 50, 60 people in this room. Nobody is look-alike to the other person here. What is interesting is that when you are looking at conventional genes, again, they are highly similar. I like to believe that these conventional genes are the key players for making the major structures. When you're looking at between mouse and human, how many eyes do they have in mice? 2. Humans have 2 eyes. Mice and humans, they share about 85% of conventional gene sequences. So these conventional genes might be critical for the mainframe construction within each species. That's why mice and humans share that level of conventional gene sequences. However, if you're looking at the rest of the genome, so conventional genes occupy about 2% of our genome. But when you're looking at the rest of the genome, we are so different. We meaning mice and humans, we are so different. And many scientists say that these non-conventional genes as a non-coding RNA or non-coding element. But there are quite some number of coding sequences in these non-conventional gene sections. So we don't know how many are there. Maybe 100,000, maybe 200,000. Conventional genes, they are talking about 20 to 30,000 here. So if we are looking at the conventional genes, highly homologous. However, if we are looking at non-conventional genes, we are so different. Even within a human species, we are so different. Obviously, between mice and humans, we are so different. So I like to believe that these non-conventional genes may play a very important role for individual-specific as well as species-specific phenotypes. So to decode that dynamic 4 to the nth genome information system, instead of looking at conventional genes only, that's on the left, we should look at the entire genome information system. But in the middle, we are still looking at dynamic genome. But if you're looking at temporal and spatial genome, and also looking at the entire element, then we'll have a better resolution of biology and medicine. So I like to go back to that conventional genes and non-conventional genes idea here. So the conventional genes will dictate macrophenotypes, 2 eyes, 1 nose, 1 mouth. However, these non-conventional genes and non-conventional gene elements can play a very important role for micro and dynamic phenotypes. There might be microdynamic sensors and actuators in these non-conventional genes and elements. So if we compare person A and B, conventional genes, they are similar. However, non-conventional genes like transposable repetitive element, they are unique for each individual. So that might be the reason why person A is more sensitive to radiation injury or the infection sensitivity with the person A, because they have unique sensors and actuators for these stressors coming to the body. I guess you heard about whole genome sequences, whole genome sequencing. So whole should be complete. Without complete, it's difficult to use the word whole. So I'd like to show some of the information that it's too early for scientists and the community to use the whole genome sequence. Same slide, after 10th anniversary of completing human genome sequence, we are still working on that. And in 2013, NIH updated mouse genome sequence. Mouse chromosome Y was 16 million nucleotides until early part of 2013. And then NIH updated to 92. So now the size of mouse chromosome Y is almost 6 times larger than the annotation in 2013. However, it is interesting that human chromosome Y was about 60 million nucleotides until 2013, but even now they are still 60 million nucleotides. However, less than half of human chromosome Y is completely sequenced as of today. And it's interesting that they were able to provide 92 million nucleotides worth of mouse chromosome Y sequence, and they can only sequence about half of human chromosome Y, even though it is a National Institute of Human Health, not the mouse health. And people are talking about whole genome sequencing by using next generation sequencing. There are 3 potential assembly scenarios of the whole genome using NGS. The first scenario is that there is a complete reference. Even though there is no complete reference, let's just assume that there's complete reference. When there's complete reference, then there might be a region that we cannot sequence using NGS. NGS only provides 100 nucleotide, about 100 nucleotide sequencing read. So that means when there's a really complex area packed with repetitive element, that 100 nucleotide read cannot do the sequencing. So even with complete reference, it cannot provide sequence here. And these light gray might be individual specific DNA or genome. That means a lot of NGS sequence read cannot be used to assemble the whole genome. So that means a lot of NGS read will go into the trash can without being used. The second scenario is that there is no complete, that means incomplete reference, like as of today. So that means there might be some gaps in the human genome. That means we will have a lot more NGS read going to the trash because there is no reference to assemble. And when there is no reference, that means there will be a lot more NGS read sequence that they cannot use to assemble whole genome sequence. So currently, since there is no completely sequenced reference, and also we don't know whether humans share 99.9% of genome sequence as of today. So that means a lot of NGS read sequence will go into the trash without being used to assemble the sequence. So therefore, the claim of generating whole genome sequence by using NGS sequencing read doesn't make much sense at all. So is it logical, scientific, or realistic to sequence whole genomes of 1 million humans? when no single reference genome is completely sequenced yet. In England, they started a new project, and they are going to sequence 1 million people. But yet, US NIH is still struggling to complete one reference for over quarter century with for more than $4 billion. So if you are looking at homologous and static genome, if you assume that humans' genome are highly homologous and static, that's fine. You can just go straight 2003, I complete the whole genome, and then I celebrate the 10th anniversary 2013, and then 1 million genome project, no problem. Give me $10,000, I'll give you the whole genome sequence. But in the real world, it's not that simple. We are highly diverse, and our genome is so dynamic. So in 2003, NIH couldn't finish it, but they did 10th anniversary of completion of the Human Genome Project anyway. So they just went ahead with that celebration with Smithsonian Museum. And then now there are many institutions claiming that they'll do 100,000 genome project. Some, like in England, they are talking about 1 million genome project. I don't know how long it will take for them to complete 1 million genome project, and I don't know how much money it will cost. Now, another assumption that I like to talk about is that we use cell lines. Then, so how do we apply all the data that we collect from cell culture-based studies to the real understanding of biology and medicine? It is a big question. I'm not against using cell lines, but we need to have this discussion. Is it really useful to collect the data from cell lines and then apply to the real life? So during the course of immortalization to make cell lines, there are some unintended changes in genotypes and phenotypes. After all, those genotypes and phenotypes of those cell lines might be quite different from the primary cells which you were intended to study. So there's a primary cell, and then immortalization. Then now we have immortalized cell line. Now you will have slightly different genome as well as the phenotype. And also, we put the cell line into the culture depending on number of passages. So this might be the first original cell line, and then with the passages, it will acquire different genome and phenotypes as we pass along. So this passage will have quite different genotype and phenotype, even from the same cell line, the original same cell line. Not to mention differences between the primary cell that were used to create the cell line and these cell lines after multiple passages. So this is one simulation here. If we study like LPS lipopolysaccharide stress, and if we stress primary cells, like a human blood cells with LPS, we may have this type of response. This is a simulation. This is not the real data. We may have this type of response. However, if we use cell lines derived from the blood cells, so now we are starting with this and multiple passages. Depending on passages, we'll see different response to LPS. Then would these data sets from the cell lines useful to understand this type of response? Then we need to discuss. So when we study the function of conventional gene, we typically focus on one primary isoform of the gene, meaning that one combination of exons or one messenger RNA type. Is it sustainable? Do we need to focus more on the multiplicity of transcripts or isoforms of a single gene? So typically, if we study conventional gene, we assume there are 1 or 2 messenger RNAs, or maybe 3 or 4 messenger RNAs formed. But in many cases, it is highly likely that a single gene can produce many, many messenger RNAs. Some we don't even know. It will be really depending on individuals. Some individuals may produce 20 different isoforms of glucocorticoid receptor. The others may produce only 10 different isoforms of glucocorticoid receptor gene. So currently, when we are talking about interactions between two genes, we are talking about protein A and protein B interactions. But what if each gene A and B produce 20, 30 different isoforms, depending on the condition, meaning time and space, or even stress can activate some of these isoforms? Then the combination is huge. So when we study the interaction between gene A and gene B, which combination would you pick out of these multiple combinations? And also there's uncertainty in detecting the combinatorial presence of multiple protein isoforms in the study of immunohistochemistry, flow cytometry, and others. So in this case, one gene can produce multiple isoforms. These are the phenotypes. Let's say this is membrane protein. And then this antibody can bind to this epitope. So this antibody will bind this isoform and this isoform. It may not bind to these three isoforms here. So let's say this is the cell that we are interested in looking for. But the antibody will bind to these isoforms, but this is not really the same as this cell. And also, there's a combinatorial expression of different isoforms on the membrane. So this antibody will bind to the right isoform. However, on this membrane, there are other isoforms expressed, and this antibody cannot bind. So even though this antibody can detect precisely that isoform, indeed, this cell is not equal to the cell of interest. Same with this. It's not going to detect this isoform here. And also, let's say there is a marker to detect a specific cell type, skin. I'm just using very rough description of skin, not the cell here. So this marker this protein marker is used for detecting skin. However, there's isoform of this protein, this gene produced, and then the same antibody can bind. But obviously, it has different genome and phenotypes. And also, there is different types of protein produced. And if we use a secondary antibody, and that antibody is specific for neuron, so even though it was picked up by this antibody against skin-specific marker, now we are looking at the, instead of the skin, now it can be neuron. So because of these multiple isoforms of a single gene, this type of antibody-based detection can mislead us so much. And also the immunohistochemistry and flow cytometry, same concerns. So with one antibody, it can detect two isoforms. Now we are counting the cells on immunohistochemistry, 25. However, indeed, this is an intended isoform, but this is not intended isoform. So the real number for the positive cells will be 15 instead of 25. Same with the flow cytometry. So with one specific antibody, it can detect two isoforms. Two isoforms will have completely different function. So n equals to 25. However, only 15 are the intended isoform, and 10 can be unintended isoforms. What do we do with this data? So currently, we focus on really simple scheme of isoforms of a single gene, but we need to consider that there are multiple isoforms of a conventional gene can be produced over time and space. So instead of doing simple biology, we need to choose that complex biology which account for the multiplicity of transcripts and proteins from a single gene. Then what is the impact of heterozygosity on functional characteristics of a conventional gene? Typically, when we study a gene, we just focus on one gene and mutation here and deletion here, and then we study the function. But we all know that there are two alleles. So there are quite some number of conventional genes with heterozygotic status. Then what's the impact of heterozygosity here? So each allele will produce multiple isoforms, and then they collaborate. Therefore, they can produce dynamic and multiple functions of a single gene. So two alleles, and then each allele can produce different isoforms with different functions. And then at different time point, there will be differential expression of each alleles. So some of them may interact together. So allele A and allele B will interact, and then the net function will be like this. But at time point 2, different combination of allele A and allele B proteins and different function. And also about the space, tissue 1 and tissue 2 and tissue 3, they will produce different functions from a single gene. So within an individual, depending on space and time, and single gene can produce two different types of proteins from allele A and allele B, and then depending on time and space, the net function will be quite different. One gene will have many, many different combinations of proteins when we account for the heterozygosity. So now the chapter A about systems assumptions finished. And then I'll move on to the chapter B, one section that is about nucleic acid-based diagnostics. So I wrote this section almost a year before this coronavirus problem. So in 2015 in South Korea, there was a major outbreak of MERS, Middle East Respiratory Syndrome coronavirus. And so after I saw that outbreak, I decided to write that section. So I realized that there's a potential inaccurate diagnosis of viral infection by PCR. So in this case, I'm using MERS-CoV as an example. The PCR reaction, I guess everybody heard about PCR. PCR cannot be perfect in terms of specificity. We have to go inside of the tube and look at the dynamics of interactions or kinetics of DNA molecules and enzymes and others. So on the top panel, I'll assume that there's 100% specificity here. So with the PCR, I'm looking at 5 different scenarios. Yes, that PCR detected the target, which caused MERS. That's good. But that PCR detected another target. However, that virus has different sequence. And indeed, it's not pathogenic. Another virus, different sequence, another not pathogenic. And also it can detect another virus, unknown specificity, because there are so many different types of coronavirus in the world. Everybody in this room, I expect that you have coronavirus. And we have different subtypes of coronavirus in our system. And since this is 100% specificity, with this diagnostic system, this virus may not be picked up. So this can be our commensal virus. Commensal meaning that we have it all the time, like a herpes virus. When we get sick, it is activated, and then we get cold sore. Similar to herpes virus, coronavirus can be activated during stress conditions, like a flu-like symptoms. If you have high fever and inflammation, it will activate your coronavirus. So if we simulate that situation, then in this case, so this is our commensal virus, which doesn't do much. This is not pathogenic. So with the current coronavirus MERS coronavirus detection system, the level is too low, and also there's some difference with the specificity. So there is no detection with a normal condition when we are not sick. However, when we have a major flu-like symptoms from the influenza virus infection, then it will stress our body, and then it will increase the copy number of this non-pathogenic coronavirus. Because of the increased copy number from the stress of flu-like symptoms, this detection kit will identify what produced the amplicons. And then it will say coronavirus positive. However, this is indeed false positive because it detected our commensal virus, which were activated from the stress of influenza virus infection. So it is likely that current system might be in similar situation. So Wuhan coronavirus, I'm not sure whether the coronavirus that we see in United States came from Wuhan, but so in your medical school, I assume, or in your college, you heard about Koch's postulate about the germ theory of disease. There are 4 steps to confirm one microbe causes this specific disease. So in this case, coronavirus caused this current epidemic of pneumonia and flu-like symptoms. The first step, all subjects manifesting the disease should have the coronavirus. I don't think we have that evidence. And the specific microbe should be isolated from the subject with the disease. Maybe they isolated. I don't have the data. Number 3, the cultured microbe must cause the specific disease. We don't have this data at all. Number 4, the microbe should be isolated from the inoculated subject with a specific disease, should be the same as the original coronavirus. We don't have this evidence at all. Even for the number 2, the specific microbe should be isolated from the subject. Since we all have coronavirus all the time, I'm not sure how they can pinpoint that Wuhan coronavirus in that isolation. It will be a mixed culture with many subtypes. So there's a potential inaccurate diagnosis of COVID-19. So identical to MERS coronavirus. So with the assumption that 100% specificity, we still have false positive. Even though they pick up nicely with the current kit, they are picking up different coronavirus, which may not be pathogenic. And same here, when people are on the flu-like symptom because of the influenza virus infection, they will have increased copy number of commensal coronavirus. Because of that, the current kit can detect this commensal coronavirus, and then it leads to false positive. So current diagnostic system using nucleic acid, protein, and others has major problems in terms of the accuracy. In other words, producing false positives. In this case, we have 1, 2, 3, 4 positive with the nucleic acid. However, indeed, this is our original target, only 1 positive, the real positive here. Same with protein, 1, 2, 3, 4 positives, but there is only 1 real positive. And microbial diagnostics, I just explained with MERS coronavirus and COVID-19. Indeed, they can detect this virus as positive. However, their detection, these 3 are false positives. We saw this slide. So every nucleotide and position in our genome information system is important for dictating our dynamic phenotypes. Again, I explained this a couple of times. In April 23, they completed human genome, and then 2013, they celebrate the 10th anniversary of researchers producing the first complete human genome sequence. It was first complete human genome sequence in 2013. This is from last week. So until the human genome, until 2013, human genome base pairs was 3.3 billion base pairs. 3.3 billion base pairs until September 2013. That was after 10th anniversary of completing human genome. September 2013 and August 2019, the human genome base pair is 4.5. There's an increase of 1.2 billion nucleotide. But yet, in 2013, they had 10th anniversary of completing human genome. How do we account for this additional 1.2 billion nucleotide? I'm not talking about million nucleotide, I'm talking about billion nucleotide. How much genetic information would you expect from this 1.2 billion nucleotide? They assumed that they completed the human genome. But now we are looking at 1.2 additional nucleotide or base pairs. So all those foundational assumptions, how sustainable are they? When human genome becomes from 3.3 billion to 4.5 billion nucleotide, are they really sustainable in accurately understanding and applying that knowledge from the studies based on those foundations? So we need to evaluate whether these foundational assumptions are sustainable or not. Reproducibility is important. However, I would say accuracy has the priority. You can reproduce inaccurate data over and over, and that's not useful. So the assumptions project will be continued, hopefully until the time when I cannot think clearly. So we like to evaluate the sustainability of scientific assumptions. And we like to bring the scientists and general public together so that we can discuss about assumptions. It may not be just in biological sciences. Down the road, we may expand to other fields. So you can find this assumptions essay, the first version of the assumptions essay on this website. You can download as a PDF or EPUB. And also, I propose to have assumptions discussions on the website. So if you are interested in joining that discussion about the assumptions, then you can just reply on the website. So that hopefully down the road, we have some assumptions discussions, at least in California, hopefully nationwide. Thank you.
Wonderful, Dr. Cho. Thank you very much for taking a very complicated topic, the human genome, and trying to explain why, even though Dr. Greenhall and I both have 2 eyes, 1 nose, 1 mouth, and gray hair, we remain quite distinct. Also, I want to congratulate you on the cartoons. I can't imagine how much work it took to make up all those cartoons, the different sequences. I'm thinking about coloring in 1 little square, and it drives me nuts. So that's also beautiful. Dave, you want to talk about how this relates to the bird research and the cortisol receptors?
Well, no, I was going to bring up another point. So, Kiho, your assumption that science should be 100% accurate. But what I like about science is that it's not accurate. The more you learn, the more you realize you don't know. If you look at quantum mechanics, now it's all statistics on where the molecules might be. And so clearly you point out errors and problems with science. I remember when I started doing science 38 years ago that we didn't know why bacteria cause us to get sick, and now they discovered cytokines, and we know now there's epigenetics, and it's getting more complicated all the time. So we don't have 100% accuracy. We realize that, and all scientists do. So to say that you're wrong, 100% wrong, doesn't necessarily mean it's correct, but we do the best we can with the tools we have. So you try to develop better tools to look at things better, just like when we first looked at stars, we looked through a little telescope, and now we're looking at galaxies billions of light years away. So we've got better tools to better understand things, but we will never really know 100% what's the correct thing. But you still have to go with the best thing. Even though Dr. Jurkovich and I have different genome, we're still humans, I think. And so doing knockout studies, they're not perfect. You need to know what problems there are. And basically, when you look at flow cytometry, we realize there's overlap and there might be the wrong genes. Doesn't necessarily mean that the conclusions of the study are wrong as long as you acknowledge that there are errors. Everything has potential error, and we realize that. So, yeah, you're just pointing out that things aren't 100% accurate, but doesn't mean science is necessarily wrong. Now, we look at the human glucocorticoid receptor, and we find things, and the first study we looked at the glucocorticoid receptor in mice, we showed the difference in the knockout animals and say, oh yeah, so things are not perfect. But you have to acknowledge that when you write your papers, and you look at the papers from the past, a lot of things we've assumed are right in the past aren't correct, and we keep changing it. And something we're talking about now, probably they'll laugh at us in the future. But it doesn't necessarily mean that science is all wrong, but you're pointing out that clearly there's differences because my hair is gray and Soman's is black, you know. My melanosomes died out, so that's kind of my comment. It says, yeah, sure, things are not perfect. There are errors in everything we do, and I don't think they're new. These are new concepts. I think, but people have to accept that there are errors in what we do.
So Dave, I agree with you. I don't expect that there's 100% accuracy of assumptions. And also, nature is so complex and dynamic. There is no way that I can claim that I'm right, you're wrong. So the whole idea of this assumptions essay is that if there's a thesis, there should be antithesis so that we can move to the next step. And this next step will be challenged by another group, and then we move to the next step. So the complexity of nature shows that without this process of challenging each other, I guess we might be just stalling. So that was the whole point.
Two questions over here, but Alicia, go first. Yell it out from over there.
Yeah. I love the microphone. Yeah. I'm not proud. So I'm in the genomics graduate group at UC Davis, and I really loved your presentation. A couple of things I wanted to emphasize and also piggyback off Dr. Greenhall is that when I'm reading a sequencing or bioinformatics paper, the very first thing I look at is what year it was. So case in point is your example of the human genome drastically improving from 2013 to now was that in that interim was the invention of the nanopore sequencing and the ability to read DNA from end to end from a single cell, which we did not have before. You also have to remember when something's published, it says we have a 99.9% completion rate. When you're talking on the order of billions, that's hundreds of thousands of base pairs that are not accounted for. So what we see clinically as a really, really good statistic when you're speaking about sequencing is not necessarily the same. The other thing I wanted to say is there was a really good paper at the end of last year regarding RNA sequencing, so transcriptomics, not genomics, but they called it RNA sequencing the teenage years, and that was speaking in respect to the software that had been developed to analyze the data, and how that is also constantly improving and constantly changing. I think where this field is going, relying on the genomics and the sequencing, is that it gives us an ability to look for questions that we didn't even know how to ask. I'm a big believer in hypothesis-driven research. I'm also a believer in the fact that when you gather this much data and you're able to explore it, that it's going to generate hypotheses that should then be validated in the lab. My fear for this field in particular is that the people and the bioinformaticians who own all this sequencing expertise are getting siloed because it's complicated to understand, and it's very technical, and there's a lot of jargon, and they're not communicating as well with the benchtop scientists and the clinicians to really target the efforts in a meaningful way. So to the young residents, I would just say that this field is wide open. We are layering technologies with sequencing at the genomic level and the proteomic level and the epigenetic level, just like the diagrams he showed how it's a lot more complicated than we think. And we really need to unsilo this if we're going to not only reduce the assumptions we're making, but translate that into meaningful clinical work. So thank you so much for your talk.
So I'll just make one quick comment about the genomics. If you go to NIH website, Dr. Eric Green, the director of National Human Genome Research Institute, clearly says that all humans share 99.9% of genome sequences. It's impossible for anybody to calculate 99.9% identity when he does not have a sequence of the single chromosome. So that's the level of the assumption that we have, and we need to challenge that.
Last question for time.
Yeah, hey, Dr. Cho, really interesting talk. NGS, next generation sequencing, is commercially available for cancer genome sequencing, and I'm wondering what is your understanding of? I'm assuming there's a lot of variation as well, even within a single solid tumor. Each cell probably expresses multiple isoforms, but is it, to your understanding, more complex in a tumor since there's constant turnover with these cells, or is there a different behavior in the like the genetic variety within tumor cells?
I mean, we all know that there's heterogeneity in cell types in the tumor samples. So the core of NGS-based genomic science is that they are looking for mutations of genes. Let's say they are looking for mutation of androgen receptor in the tumor sample. I just simplified it. But there are many, many genes which have pseudogenes. Pseudogenes meaning that there is original target here and multiple locations of pseudogenes. And pseudogenes are typically formed by the exons of these original genes. So when you are looking at NGS data set, like an NGS reads about 100 nucleotide, when you have the 100 nucleotide with point mutation, there is no way that whether you can confirm that that is from that target gene or that target gene's pseudogenes. So it will be misleading for the tumor scientists when they interpret that data set. So the whole idea of this assumptions project is that we have to be very transparent and we have to have open discussion so that people who are not familiar with those techniques can evaluate with their own ideas and experimental designs.
All right, thank you very much. Time's over. We got to go.